He worked previously in pharmaceutical industry research, in drug development and approval, and he is a director of the translational program of the Oxford Translational Myeloma Center.
Thank you very much. Thanks for the kind introduction.
We'll just pause 30 seconds. Lots of seats towards the front and the sides. Those who are loitering at the back, please do come in.
Shall I wait? Okay. Well, thank you very much for the invitation to speak here. I am relatively new to the UK myeloma community, but I've been involved in myeloma work for a while, at least the last 10, 12 years. And so I'm going to really talk about my experience in learning, but I'm going to start with this slide because somebody came to see me last week, and until then I actually didn't know what I will talk about, to be absolutely honest. Then this young clinical scientist presented this experiment and said she wanted to do this because she wanted to find out what is written on the right-hand side, the biological predictor of resistance and sensitivity in patients. And I gave her quite a long rant about why you should not do this experiment. Or if you do, you should be prepared what sorts of answer you're likely to get. It may not be what you're thinking you're going to get. So this is going to be a slightly longer version of what I was telling her, don't do it. But I now present some data and see if I can convince some of you, at least some of you, that this experiment needs to be thought carefully before you actually spend the time and money to do it. So LEN and POM, lenalidomide, pomalidomide, are the immunomodulatory class of drugs, and the top three, as you know, are approved in multiple myeloma treatment. The bottom two are also IMiDs, but they're branded differently, and if you have a question, I can tell you why. This is an IMiD. Everything is an IMiD. Everything has a, the glutaramide ring is considered an IMiD, okay? And that's the part that is critical for targeting all these drugs. These bottom two are actually being developed in myeloma. Both of them are being developed in myeloma, and I was one of the earlier scientists to actually try to convince the company that we could produce more of these drugs in myeloma. Now, the beauty of this is that all of these drugs actually, or compounds, actually target one protein as the primary mediator of their activities. Now, I don't know about you, I have not seen anywhere that you could successively target multiple different drugs to a single common protein target, and successfully develop drugs that are approved and used all over. Revlimid, you probably know, is the annual sale of close to $20 billion per year at the peak sale, and POM close to $3 billion. Now, there are atomic difference between two drugs. One atom difference. The next generation ones are slightly more complicated, but they are essentially the same category. Now, the key to all of this was 2010, when the target of thalidomide was actually discovered by a Japanese scientist group along with Celgene and San Diego. And it really opened up our thinking about these class of drugs. Now, cereblon is part of a E3 ligase complex. It is the protein that actually brings the substrate proteins to the E3 ligase for putting ubiquitin tag. And so that led to a development of a molecular mechanism of this class of drugs. Essentially what happens is binding of the drug to cereblon within the context of the E3 ligase will then bring substrate proteins, specific substrate proteins. Here we are showing 3 of those, Aiolos, Icaros, and ZFP91. And then after they're tagged, they're degraded by proteasomal processes. Now, that has been the classic mechanistic model of all of these molecules. And even now, it has not changed much. Now, the thing about this is that we could see two different kinds of activities based on this basic molecular mechanism. One was going to be in the, what happens in the myeloma cells, the cell autonomous effect, and where the effects are typically a reducing proliferation and increasing apoptosis. In the immune cells, the same mechanism apparently would lead to immunostimulatory activities via different cytokines. Now, these are, the substrates are transcriptional repressors, and so it's a de-repression mechanism and activation of different kinds of transcription. Now, this story for Revlimid and pomalidomide really created this bimodal, or rather two different kinds of mode of action of these drugs. One is the tumoricidal activity, and the other is immunomodulatory. And then, if you think about combinations across all the different type of myeloma therapies, you could generate data to show that there's synergistic combination of this class of drugs with other myeloma agents. On the immune side also, it's extensively studied for various combinations, but there is a general trend of anti-myeloma activity and then immunostimulatory activity. And those are sort of available in so many different trial combinations that you have to spend a day to figure out in individual combinations. Okay, so in 2016, before we were trying to develop the so-called cell mods, I was asked to essentially help the company think, how would we go about developing new molecules when you already have 3 drugs approved, and what's the difference? How would you even take the development path? So I created this slide, and at the time, I presented this view that we do not know if there are other targets, but if cereblon is the only target, then I could imagine a scenario that different drugs could exploit a different cluster of proteins, substrates. Some of them could be common to all, some could be partially common, but there could be unique substrate proteins. And then, based on the kinetics of degradation of those substrate proteins, you might actually elicit different type of phenotypic response. Now, there are a whole host of different type of immune cells, and you can imagine that there will be some differentiated activities in those cells, and as a result of that, you might have differentiation of these drugs. I have, sorry to say, that so far, we are at the very surface of this problem. And you will see that I'll show data on cereblon mainly to kind of convince you how challenging and difficult this can be to sort out the problems of this class of drug, especially when its molecular mechanism is so narrowly defined. We have not been able to identify additional substrates of consequence for multiple myeloma. So we are all stuck with the 2 key substrates that we know, which ALOs and Icaros, and we try to explain the behavior of all these drugs to those 2 substrates. However, around 2015, one of the things we did was to look at the kinetics of each of those drugs, and we profiled them, and Chad Brooklyn in the lab actually did this work very painstakingly, and that was one of the ways we thought the cell mods will actually work. So if you look at iberdomide or mezigdomide, the 2 cell mods in development, they are more potent binders of the target, but they are also much more fast in terms of turnover of the ALOs and Icaros and downstream IRF4 MYC that we used. And this is one of the screening assay for iberdomide. This is not published. So this leads to my learning number 1 that I wanted to share with the younger folks. There was a pre-cereblon world where IMiD drugs were thought to be promiscuous in their mechanism. What it meant was that based on all the different type of cells, there'll be different proteins that these drugs will bind and work with, because they didn't know there was a single target. So the idea was that if in myeloma cell it'd be one way, in immune cell, NK cells could be different, and megakaryocytes could be different, B cells could be different. So we had this promiscuous view. The cereblon discovery narrowed that thing. So actually, you have one target, but the effect would be different in different cell types. That is the pleiotropic mechanism. But then, the more molecular stuff we learn, now our view has been much more precise. So one of the things about the cell mods that is different from the IMiDs is cell mods are engineered out of a fairly large library of 2.5, 3 million. Do you know how many compounds were actually screened to get lenalidomide, thalidomide, and pomalidomide, the initial library is actually 3,000. And there was another drug that actually we didn't develop that came from this 3,000 molecules is the original source. And the cell mods, and now it is much larger for other reasons because we can develop substrate-specific imids as well. So for a scientist, it's actually an excitement, but it's a damn challenging when you have to differentiate all these chemical matter against a common target. So how do you differentiate it? I will tell you understanding of cereblon now. So cereblon loss and mutations. So when we started, it's about 2012. There was a scientist in Celgene, Antonio, and what Antonio was doing was to develop a resistant cell line in the lab. And this is a case where she developed H929, is quite common myeloma cell line, and with increasing amount of lenalidomide, and she produced 4 different subclones of H929, which were resistant to len. Okay. Around the same time, Keith Stewart group published a paper showing that in various cell lines, the amount of cereblon could be different. That it was not like a well expressed across all the different cell lines. Some cell lines had more, some had less. And this led to some amount of anguish in our company because there was a thought that if the level of cereblon in patient was not enough, high enough, whether those patients should get treatment with IMiDs. The target is not there, why should you give the patient the drug? So that was a concern. So I got into the game and tried to understand exactly how we define this. So first thing I did was sequence cereblon gene from newly diagnosed patients and cell lines and to see are there mutations in this gene that could make them unstable, or will there be a general presence of a diverse mutation range in this gene? Turns out that I didn't find any. It's very rare in the newly diagnosed patients. By the way, cereblon is not really part of a myeloma biology. If you delete cereblon in a myeloma cell, nothing happens. It actually gets almost like brought into play by the induction of the drug binding. So it's almost like a neomorphic biology that does not exist in nature, because we do not know the natural binder of cereblon, a small molecule binder. We do not know. So the drug is doing something quite interesting. Okay. Now, what Antonia saw was that this cell line, there was a partial activity of pomalidomide while they were resistant to lenalidomide. So that was interesting. So the thought was whether pomalidomide could actually work with less cereblon in the cell. So its requirement, now potency is very similar by the way. The binding affinity to cereblon is not that different. However, it is able to catalyze the reactions or have the kinetics faster than lenalidomide. Maybe that's how it is working. Subsequent to this, another few years later, we tried to do the same experiment, but this time with iberdomide. So Chad in the lab, I asked him to look at various POM-resistant cell lines, and the question I asked was, do you see any correlation between the level of cereblon and whether iberdomide works or doesn't work? So this is sort of a graphical representation of the data. As you can see, there is a variety of cereblon levels for resistance to POM, the blue bar. And then the green shows that the partial activity of iberdomide in those. So it actually varies. There's no clear correlation between level of cereblon versus the activity of iber. The right-hand side shows sort of the biochemistry of it. If you look on the top, this is the degradation of ALOs in sensitive cells versus POM-resistant cells. POM at 1 micromolar doesn't do much. And you can see that iber at 1/10 the concentration of pomalidomide is able to catalyze the degradation. Okay, so going to the next stage, this is immunohistochemistry. We wanted to actually create an assay whereby we can look at protein level. And this is actually an immunohistochemical assay developed in the San Diego lab. And you're looking at expression of cereblon measured through a monoclonal antibody the sensitive cells versus resistant cell. This resistant cell actually does not express any cereblon, and therefore you don't detect it. So taking this type of assay, this is a collaboration with Niels van de Donk's group, where he had patients that had refractory to lenalidomide. So we had samples, and we analyzed them by looking pre-post cereblon level. And what you see here is between diagnosis and when they're len refractory, in a majority of the patients, the protein level of cereblon went down. But there are a few cases actually see increase as well, and some cases it's relatively stable. So this is something that we can measure in patients, and we see some loss, but sometimes we see the amount is not really changing much. Okay. What about outcome? I'm sorry, this is tiny. So this is cereblon measured by immunohistochemistry. This is Peter Sonneval's Hoven 87, and he looked at, I think, about 500-600 newly diagnosed patients, and then immunohistochemical analysis of cereblon level. And what he found was a fairly nice correlation to higher levels of cereblon giving better PFS and OS. We did the same thing now in relapse patients, but this time looking at gene expression level. So Max Chen, a scientist in my group, and this is around the time of pomalidomide approval, we had to do this, looked at expression of cereblon, and what you see up there is across close to 250 patients, we are trying to understand correlation of response to levels of cereblon. As you can see that even patients with very low cereblon level, you can have partial response, and you can have stable disease, or you can have progressive disease. It didn't matter. Very high cereblon level, some patients progress through. So, and then next to that, we try to see if there's an outcome effect. And in this case, even though there's a separation, it was not statistically significant. All right. Then, unfortunately now, Sarah Gooding joins the work. So fun stuff will happen. So we were doing a myeloma genome project in the company. We did a big myeloma genome project to newly diagnosed patients with Gareth Morgan and Nikhil Munshi and a few others. But this is an in-house project that I was running with relapsed patients. And I had a collection of trials and samples collected. So we did a genome analysis of, I think now it is up to 600-700 patients, but at the time we had like 400 so. And so what we wanted to do is to understand the landscape of relapsed myeloma. And so the way we went about doing that is to really compare with about 200 newly diagnosed against 325 patients. Some of them are refractory to LEN, some to POM as well, and then generated whole genome sequencing and RNA-seq data. Okay. Several types of questions were asked out of the data. So the first one was cereblon. Now, when I initially analyzed cereblon gene form, the mutations were very rare. But in this data set, what Sarah was trying to do is to categorize what are the different types of cereblon changes we can identify. And it turns out that there was a fairly large amount of cereblon gene copy loss. There were structural variants, like inversions with cereblon. And then this character, it's a transcript. It's an isoform, which is a 9-11 junction with a deletion of exon 10. We actually originally identified in CLL. We never looked at it in myeloma. But with larger data set, we began to look at del10. And Nizar and I have been doing this for a while now to figure out what does del10 do. I really don't know. But leave that aside, Sarah found out that this is the distribution of the cereblon dysregulation in the POM in the LEN category up top, and then in the POM category. The bottom line is it increases as patients go through these treatments, and there is a general increase of cereblon abnormalities. Then this one is a slightly more interesting view of the structural changes that would be translocation, deletion, mainly deletion, and inversion. And as you can see that these patients actually grow in these abnormalities with time and treatment, but they become more clonal as they go through to later lines. The right-hand side is actually another way to kind of describe the importance of this copy loss. What you're seeing is across the genome, every 5 megabase, we're trying to understand the landscape of gain and loss. And across that, frame, you see cereblon is at the very high here. 3p loss is very dominant. The closest to that is the 17p TP53. So it's actually very marked in myeloma patients as a part of the genome. It's the 3p that is so much affected. Okay. The other side of the story that Sarah was actually able to follow was based on, sorry, this is to show the increase of the exon 10 splice variant, which I mentioned before, and you see that this also increases with treatment. Now, I added a question mark here, because despite predicting that there will be a protein, it's just an exon skipping event. It should produce a 23 amino acid shorter cereblon, and that should not have the drug binding moiety. But we have never been able to identify it. I spent a year trying to detect this protein. It's not easy, but I do not know if there is this protein. It doesn't appear in cell lines, and there is no way to know if this is actually important. However, the transcript, the exon variant in later line patients is much more than we see in the newly diagnosed. So there is some kind of an effect of delta, but somebody needs to work out what that is. Okay, the other side of the story Sarah was chasing is this regulators of cereblon. So there is a complex process by which cereblon is maintained. So this is actually a nadylation, denadylation cascade to make inactive E3 ligase, cereblon E3 ligase, so that it doesn't actually destroy itself. And that is mediated by the denadylation is mediated by the COPS9 complex. So long story short, we actually started to look for those, and if you read her paper, she describes how she did it by identifying sort of most of the citations on these, and then some criteria we used, etc. But long story short, we ended up with sort of 3 areas. 3p was already known. This is the cereblon part. But then 2q is the new one that we found, and the COPS genes are all there. So this is actually representation of the data that we have on COPS loss in LANE and POM. Similar to what I described for cereblon, now you see regulators of cereblon is also getting affected by copy loss. Then it does have an effect on the expression level, all right? So to end that part, what is the outcome? So it turns out, if you take all of these different defects of cereblon separately or together, as one or more, there is a clear effect on PFS. And for DEL10, we also did a comparison with the newly diagnosed, and turns out it's not too many patients we could find, but there was an effect that we could see there as well. Okay, is there any cereblon independent immune resistance mechanism? This actually I'm showing a paper we published from the genomic landscape work, where we see enrichment of, these are significant players in myeloma. You will recognize that there's an enrichment from LANE to POM, and the frequency increases. We also looked at double hit events, and the clinicians are extremely interested, Martin particularly, the double hit myelomas of various kinds, and what is the prognosis of them, what type of treatment we should give. So we see enrichment of various double hits, and I think they might mean something worse for the disease progression, but whether or not this is connected to cereblon is still not resolved. I used to think they're independent, but I have revised my view. So I will not clearly state this is cereblon independent. I think there could be a very clear interaction between high-risk disease markers and cereblon, at least in our data set. So you have to wait to figure out what that is. Maybe it'll take some more time. Okay, I want to end, I have 5 more minutes, so I'm going to end with this last a few slides on iberdomide. And this is the last trial I managed when I was in BMS. And this is to clinically develop iberdomide. This is a phase 1 trial, and I think at the time we wrote the paper, we had 200 plus patients in it. And the analysis is looking at cereblon level and response. And this is the part that really I find interesting. These are median 5 lines of treatment into the phase 1 trial. So many patients could be 7, 8, 9. There was not a single patient we found who lost cereblon completely. So unlike your cell lines, in patients, it's very hard to find 0 cereblon. There's some expression of cereblon, and I will show you a clinical case. And on this side, you see the correlation, response, and doses. And this is a phase 1 trial, so you're trying to figure out there's a dose-based response, but there's no such correlation. Responders and non-responders are similar distribution, not really that different. Now, we combine all the things we learned from Sarah's work and in the relapsed genome work. We try to figure out in the patients that we have, how many actually had cereblon-related changes. And this is actually the list. I think we had total, how many? 34 with dysregulated cereblon and 48 without. So we checked out the response to iberdomide in this category of patients. So this is what you see, sort of top is wild type and these various kinds of dysregulated cereblon. So across the board, there was some activity of iberdomide. It's not stellar. The overall response is 30%. So only 3 out of 10 would respond. But what is interesting, and I think that's the part that I found most profound, is that patients that are wild type or cereblon dysregulated, if they respond, they have very high PFS. Non-responders are very similar, but so if you are a cereblon mutant or a cereblon deleted or del10 or whatever, if that patient has responded to iberdomide, this is iberdex, by the way, then that completely overcome whatever is the inadequacy mediated by them. So that I found to be quite fascinating. So we published this. We need more data to understand the nature of response. But I'm going to close with this slide. And this is the second lesson, that understanding response and resistance could be complicated. Therefore, that first experiment, don't jump into it without thinking. So you have on the left the patient. This is 1.6 milligram dose, multiple cereblon defects. The response was a VGPR. Duration was 10.8, and PFS is 11 months. It's 4 times more than the general population. This is a responder. What is happening in the bone marrow? In immunohistochemistry, you can see the Aiolos level is going down marginally, not much. This patient, we didn't have Icaros, so there's no that data. So immune stimulation in PBMC, we see some immunostimulatory activity measured. We did cytoph in the bone marrow as well. I completely skipped the immune story because you only gave me 30 minutes. So that could be the next one. Second patient, you have cereblon defect, and you have quite a nice degradation of the substrates for Aiolos, but not much for Icaros. 0 immunostimulation I could observe. Guess what? The PFS is 19 months. Okay? The third patient, very nice degradation of substrate. This Aiolos substrate, not Icaros. Then you have some immunostimulation, then you have very nice response. One thing I forgot to tell you is, I think one patient, the middle one, this one, cereblon level is actually very, very low. The score goes from 0 to 300, and this is 19. So you can imagine that you could have fairly decent degradation of the substrate even with such low cereblon. So what's going on? It is complicated. So that's the message. So before people run out of the room, let me conclude with the summary that I think it is fairly well known the cereblon is the key target of this class of proteins. And clinical resistance could be due to loss of cereblon by expression, mutation in isoform expression. You could have additional cereblon-related proteins like COPS. And there could be still cereblon-independent mechanisms, purely disease-driven mechanisms that could be there. That could be genetic, epigenetic, or immune. I think we didn't get time to talk about immune scenario for resistance. That's actually extremely exciting area. POM can rescue LEN resistance, and IBER can rescue POM resistance. That's interesting. What does MESI do? We don't know yet. So we have to wait to see what mezigdomide does. And the last point is clinical response and resistance is multifactorial. It is not easily understood by looking at one lens. I tried various kinds of things, but it still seems not adequate enough to give a complete and satisfactory answer. So I'm back to this slide. Are we going to do this experiment or not? I am not spending my time. So this is to acknowledge my colleagues at Celgene, BMS, Oxford, and several collaborators whose work was cited here. I didn't want to include everybody's name to offend them. Anyway, thank you. Do I take questions or none? Okay.
Any questions?